The Crustacea form a subphylum within the arthropods, with at least 250,000 species known worldwide, which are characterized by a huge diversity of shapes and evolutionary adaptations to different habitats and ways of life. That diversity can make it difficult to differentiate crustaceans from other groups. The most obvious differences of many crustaceans to the hexapoda and myriapoda are the number of antennae, namely 2 pairs, and the gills. However, these features are plesiomorphies, that is, features that they inherited from the ancestral form and that have only been lost secondarily in the other groups. It also should be emphasized that the hexapoda, or insects, are phylogenetically part of the crustaceans, so they could be considered as a group of highly specialized crustaceans. But because they are morphologically so different, we will treat insects in a different video. The possession of a typical biramous limb in the crustaceans is probably an ancestral character of arthropods, since the trilobites, once hugely diverse but today extinct, also had them, but probably were not closely related to crustaceans. What remains in common in almost all crustaceans is the special shape of the larva, the nauplius, with typically 3 extremity-bearing segments and the typical nauplius eye. The development is similar in most groups within Crustacea. They usually go through several larval stages in which new segments and the associated extremities are added in succession. All Crustacea, with the exception of tongueworms, form the nauplius larva, typical of crustaceans, as the first larval stage. However, this stage can also take place in the egg. From this stage, different types of larvae then form within the various groups, for example, the zoea larvae, which then grow into adult crustaceans with or without metamorphosis. The zoea is a larval form of crustaceans that usually follows the stage of the nauplius larva. It occurs in most decapods. Here, the zoea hatches directly from the egg. The zoea is divided into the cephalothorax and the pleon, the abdomen, and, in contrast to the nauplius larva, which has only a single eye, has a pair of complex eyes. The cephalothorax has several pairs of biramous limbs. Traditionally, there are 6 classes of Crustacea: the Remipedia, Cephalocardia, the Brachiopoda, the Malacostraca, the Ostracoda, and the Maxillopoda, the taxonomic status of the latter being quite controversial. The common water flea is a species from the genus Daphnia. The Daphnia belong to the Phylopoda, the leaf crabs, and within them to the Cladocera. The body is enclosed in a bilobed shell, the carapace, which only leaves the ventrally bent head with the powerful rowing antennae free. Both shell halves merge dorsally and form a keel, which ends in the apical spine at the rear end. The rowing antennae are the most noteworthy of the extremities. These are the second antennae. The first antennae are much smaller sensory organs. A pair of strong mandibles and much weaker first maxillae have developed on the mouth limbs, while the second maxillae have been lost almost without a trace. Behind them, there are 5 pairs of legs that overlap and are covered by the shell. The legs do not serve for locomotion as they would have done ancestrally. This work is instead done by the second antennae, Rather, they serve two other functions, respiration and food acquisition. The abdomen, or pleon, is strongly curved ventrally, very mobile, and ends in two abdominal claws that point backwards. Of the internal organs, the first thing that catches the eye is the lively, pulsating heart, a dorsally lying round bag with an opening, the ostium, on each side, through which the blood enters. The unpaired complex eye attaches at the anterior of the brain. In embryos, it starts as a pair of eyes, but these merge into one eye as the animal matures. A pigment spot that lies against the cerebral ganglion is the so-called secondary eye. Daphnia reproduce mostly asexually, that is, via parthenogenesis. That means that most Daphnia are females. If the environmental conditions in their habitat change negatively, for instance due to drought, cold, shorter days, lack of food, or increased toxin concentrations, males are also born and fertilize the sexual eggs of the females. Under these conditions, up to 30% of individuals can be males. This kind of generational change is called heterogony. The European crayfish, Astacus astacus, is a representative of the Malacostraca and belongs to the order Decapoda. The Malacostraca are characterized by a largely constant number of segments, 20 or 21. The Decapoda have converted the first three pairs of theropods into jaws, the so-called maxillipeds, so that only the 5 following pairs of periopods remain as walking legs. The body is surrounded by a cuticle in which calcium salts are stored, the so-called carapace. It serves as an exoskeleton for protection and for the connection of the muscles. The European crayfish is the largest of the freshwater crustaceans native to Europe. It can reach between 15 and 20 years of age. The scientific name is derived from the ancient Greek astrakos, which means crayfish. This species can reach up to 20 centimeters from head to tail tip and can weigh up to 350 grams. There are 2 pairs of horn-like structures around the eyes that protect them. The European crayfish has 2 claws, or chelipeds, that are used to hold prey and for defense. The body is usually dark brown to reddish brown, but some individuals can be blue. The head is pointed. The undersides of the claws and the joints are red, which is an important distinguishing feature from other crayfish species. Mating takes place when the water temperature drops in autumn. The males hold the females with their claws and turn them on their backs or on their side. The European crayfish is currently suffering from the development of waterways and pollution of watercourses, but the greatest threat to it is a pathogenic water mold causing a disease called the crayfish plague. This pathogen was brought into Europe by introduced American crayfish, such as the signal crayfish or spiny cheek crayfish, which act as a vector for the pathogen, but unlike the European crayfish, the American species only rarely die from it. The competitive superiority of these newly introduced species has further aggravated the situation and decimated the European crayfish so extremely that in almost all European countries it can only be found in the wild in a few isolated locations. The specimens dissected here originated from a commercial breeding farm. The body consists of 2 main sections, the cephalothorax, formed from the fusion of the head and the thorax, and the abdomen, or pleon. The pleon consists of 6 fully developed abdominal segments. On the ventral side, the biramus segmented limbs are particularly noticeable. Including the antennae, there are a total of 19 pairs of extremities. For the dissection of the crayfish, we use fine scissors. In order to see the internal organization, the dorsal carapace must first be removed. Then, with very flat scissors, two parallel cuts are made approximately in the area of the delicate longitudinal furrows from the posterior wall of the cephalothorax forward to the level of the eyes, where they are connected by a short diagonal incision. The middle section of the carapace can now be gripped and removed with your fingers or with fine tweezers. The carapace can be detached from its base with scissors or a scalpel. If you work carefully, the epidermis will be preserved and is visible as a delicate pigmentation of the skin covering the organs. The carapace of the pleon can now also be removed with 3 cuts. Finally, the 2 sides of the cephalothorax are removed. The subsequent preparation is then demonstrated underwater. Here, we are dissecting without water to avoid reflection in the video. Removing the side panels of the cephalothorax, called branchiostegites, exposes the gills. The carapace is attached to the back in a median strip that corresponds to the width of the actual thorax, but arches freely over the gills on either side, forming 2 gill cavities. These gaps are used for the outflow of the respiratory water. The inflow takes place through 7 ventral openings, the first of which is in front of the second maxilliped, and the last in front of the 5th walking leg. There are 18 crystal clear, bushy gills in each of the 2 respiratory cavities. Only one vessel branches off caudally, the abdominal artery, or posterior aorta. It lies dorsally to the intestine and in each segment has paired lateral branches. The epidermis can now be carefully separated with scissors and fine tweezers. Most of the internal organs are now visible. The heart lies medially, just in front of the posterior edge of the cephalothorax. It is rhombic in shape and has 3 pairs of openings called ostia, of which only the dorsally located pair can be seen. The large stomach lies anteriorly. It is flanked by the 2 strong mandible muscles, which were separated from the original position when the carapace was removed. In female animals, the ovaries are fused posteriorly. A short oviduct runs from the ovaries on each side to the coxa of the 3rd pair of periopods. To the side of the stomach and the mandible muscles lie the rostral parts of the slightly brownish midgut glands, which extend backwards to the heart. The sides of the specimen are formed of the delicate, regularly arranged gills. The midgut glands are made up of a large number of thin tubes, which are arranged in lobes and open into 3 outlets on each side. These 3 courses combine to form a main course. The midgut gland's task is to produce enzymes for digestion in the stomach, the absorption of broken down food, and the storage of fat and glycogen. The stomach is divided into 2 parts. In the front, the spacious cardiac chamber, or gizzard, has two strong serrated cuticular ridges protruding into it from both sides, and another unpaired ridge in the middle of the stomach. Together with highly developed specialized muscles, these three stomach teeth form the gastric mill and are used to chop and grind the food, which is also broken down by the digestive enzymes of the midgut gland that are carried here. Sometimes gastroliths are located in two lateral outwardly protruding bulges of the cardiac chamber. These are semicircular white deposits of calcium carbonate that are used up in the molting process where the carapace is renewed. In the second part of the stomach, the pyloric chamber, filtration occurs. It contains complex folds and filters. These are used to separate coarse and fine food particles. Only the finest particles reach the end of the digestive system in the midgut gland. The coarser ones are conveyed into the rectum via a funnel valve. Including the antennae, the crayfish has a total of 19 pairs of segmented limbs. These can now be dissected on one side from bottom to top or from top to bottom with fine scissors. The first antenna, also called the antennule, consists of three consecutive trunk segments on which two delicate articulated filaments are attached. The outer filament is slightly thicker and longer than the inner one. On the outer filament there are sensory bristles from the seventh to the penultimate segment, which are used for chemoreception. The equilibrium organ, the statocyst, is located in the first trunk segment. The second antenna, the most important tactile organ of crustaceans, is much larger than the first. There are excretory organs near the base of the antenna that are visible as yellowish bumps. In addition to the long filament, there is a terminal segment that has a broad triangular scale. This is the exopodite. The next extremities are the mandibles. They have a massive, internally serrated chewing surface and a three-part palpus. The mandibles lie to the right and left of the mouth opening, which is bounded by an unpaired, transversely oval and laterally bristled plate, the labrum of the upper lip. A membranous fold, called the labium, functions as the rear closure of the mouth, opening the lower lip. This is followed by the two pairs of maxillae, which are much more delicate than the mandibles and have leaf-shaped teeth on the inside. In the second maxilla, the exopodite is noticeable as an elongated, somewhat curved plate. In living animals, it moves constantly and generates the flow of respiratory water that pulls through the gill cavity from back to front. Immediately after the second maxilla, there are three pairs of maxillipeds, in which the biramous nature of the limbs becomes increasingly apparent from first to the third limb. The exopodites of the mandibular feet are designed as feathered, flexible filaments, the hair of which can be spread. They are noticeable in living animals because of their striking activity that creates a current of water. With this mechanism, they can whirl in small food particles or fan out fragrances. The maxillipeds are followed by 5 pairs of walking legs, the periopods, acting as thoracic limbs which give the order their name, Decapoda. The first foot, called a cheliped, is equipped with a large and powerful claw. The second and third with small pincers. The claw or pincer is made by an extension of the penultimate limb segment beyond the point of the attachment of the distalmost limb segment. The walking legs are followed by the legs of the abdomen, of which there are 5 pairs. In females, there are only 4 pairs of pleopods. With the exception of the first, the ancestral biramous limb is again evident. The pleopods help with swimming, and are also used by females to carry the fertilized eggs and embryos. In the male, the 2 foremost pairs are transformed into mating organs. As sperm emerges from the genital opening at the base of the last pectoral foot and begins rapidly hardening, it is formed by these specialized limbs into elongated spermatophores, which they stick to the female genital opening located at the base of the third walking leg. In the female, the first pair of pleopods is regressed. The extremities of the last segment, the uropods, are broad plates that form the size of the tail fan. Their outer branches are biramus. The middle plate of the tail fan is the telson, which bears the anus as a clear longitudinal slit on the underside. Now we prepare a male crayfish, like the female before it. In the male crayfish, we can see white tubes that lie a little behind the heart and then run into the body. These are the outlets of the two testes, the vasa differentia, which in turn open out into the coxa of the fifth pair of periopods. The testes themselves lie just anterior and ventral to the heart and are fused together posteriorly.
It's a long way from up the Yorkshires. It's a long way to us. It's a long way from up the Yorkshires to the meanest human cause. Well, it's goodbye to fins and gill slits, and welcome lungs and hair. It's a long, long way from